Yes, I'm fine. So all this data I'm going to present is all published. So there is no concern about, so yeah, feel free to record the talk. Yeah, yes. Thank you so much for the kind introduction. So, and thank you for having me here to talk a little bit about what we do in the lab. So today I'm going to present you some of the main research lines running in my lab right now. Specifically, I will be talking about this novel role of ataxin-1 that is classically associated with SCA1, but I will describe you a novel function in the context of multiple sclerosis. So just one slide about me, my background. I'm a molecular biologist by training, but then I started working on neuroscience for my PhD. And when I moved to US for my postdoctoral training, that's where I started working on multiple sclerosis. So this was one of the projects during my last postdoc in San Francisco that I brought with me here at ECU. So I will start this talk just giving a brief background on multiple sclerosis for those of you who are not familiar with this disease. So this is a very common neurological disease. It's actually the first cause of non-traumatic brain injury among young adults, people between 20 and 40 years of age. So it's characterized by the loss of myelin, that is this fatty sheet that covers the axon in the central nervous system. And the loss of myelin is caused by the autoimmune attack. We still don't know exactly the cause of this disease, but we know that it causes a very broad spectrum of neurological symptoms, some more benign, some more severe. And also we know that it's beyond loss of myelin, we also observe at some point neurodegeneration that cause a lot of cognitive and motor function deficits. So as I said before, we still don't know exactly what causes MS, but multiple models have been proposed. So here I'm showing you two of the most common models. So on top of this slide, we see the outside-in model, where the trigger starts in the periphery. So something activates the immune system that migrates toward the CNS and cause neuroinflammation and eventually neurodegeneration. On the bottom, we see the inside-out model. This is basically the opposite. There is some kind of neurodegenerative event in the central nervous system that causes the death of neuronal cells, which spread epitopes that cause the activation of the immune response. And only in a secondary moment, there is the migration of these cells, of immune cells, into the CNS. So we still don't know which one of these models is true. We have data in support of both models, and maybe that also there could be both of them coexist, and the cause is more complex. Other information about MS is this a little bit of epidemiology of the disease. Is more common in European population. The incidence has been increasing in the last century, but we still observe a female to male ratio of 3:1. The average age of onset is between 20 and 40 years of age. And we also see a strong influence of latitude and ancestry on MS risk. And also there is also family history about this disease. Specifically, this evidence that there is a family history in around 20% of MS cases led the MS research community to hypothesize that MS is a genetic disease, or at least as a genetic component. But before talking a little more in detail about the genetics of MS, that would be the main focus of the talk, I just want to briefly describe the clinical evolution of the disease. This is very typical. So we observe as many diseases, they have a pre-symptomatic stage, but after disease onset, we observe what we call the relapsing remitting MS, in which we see mostly inflammation in the brain, and we have these peaks of disability followed by complete or partial recovery. And this stage can last for several years, up to 10, 15. At some point, although, the disease converts to a secondary progress, where we see much less inflammation and mostly neurodegeneration, and we observe the constant irreversible accumulation of cognitive disability. We have, I would say, around 15, 20 treatments to control the relapsing remitting phase, but still we have no effective treatment for the secondary progressive. That now is the focus in the international community. So as I mentioned, MS is a genetic disease because we see that a few epidemiological evidence is more common in relatives, and also there is a concordance in monozygotic twins compared to dizygotic twins and non-twin siblings. In particular, if we plot the lifetime risk of developing MS versus the shared genetics, we observe that the monozygotic twins, they share the maximum of the risk, up to 35%, around, while the general population is less than 5%. This clearly shows that there is a genetic component. But the fact that the monozygotic twins don't have 100% of risk means that the genetics is not the only component of this disease. So what we think is MS belongs to the so-called complex disease family. Basically, it's a polygenic inheritance. We see multiple allelic variants common in the population, each one contributing a small portion of the overall risk. As I said, MS is not only a genetic disease, but it has a genetic component. We believe that also we have an influence of the environment, such as smoking, diet, sun exposure, specific pathogens, such as viruses. And we also see epigenetic mechanisms contributing to MS risk. So we see that these three main components, genetics, epigenetics, and environment, the interplay between all of them determines overall the risk of a person to develop in his lifetime MS. So how do we study these complex diseases where we have multiple alleles, multiple genetic variants? Classic genetics is not a feasible approach. Is not powerful enough. So the research community has developed this new paradigm that is called GWAS, genome-wide association studies. Basically, there are large cohorts of cases and controls not just families. They are compared for the uneven distribution of specific allelic genetic markers. The most common are SNPs, single nucleotide polymorphisms, and they are associated to specific traits, in this case, MS risk. Through this approach, we can scan basically the genome for specific portion of the DNA that are associated with the disease. So in the last 20 years, there's been a huge effort from the international MS community. Indeed, they developed this IMSGC, this International MS Genetics Consortium, where multiple labs around the world, and here you see one of the last pictures of this consortium, put together expertise, samples, data to actually improve the power of this GWAS approach. And this is what we call the 2023 MS Genetic Atlas. So what we basically know so far about genetics of MS. We know that after 15 years of this GWAS screening, more than 200 genetic variants have been associated to MS risk. Most of them are on autosomes, but also we also found one genetic variant on the X chromosome. Overall, they explain about half of the whole genetic risk, and we think that still are some missing variants that have smaller effect that might be found in the future, increasing the sample size. So the talk of today stems from this atlas, this last picture. In particular, so I should mention that this GWAS approach is a powerful technique, but only tells you a statistical association of a specific genetic variant to the disease. It doesn't say anything about the biology behind this association. So there is a lot of effort to follow up with the GWAS data to understand what is the biological mechanisms beyond this association. So during my postdoc, I focused on one of these 233 variants that maps to the chromosome 6. And in particular, I'm showing here a typical plot where you see on the y-axis there are peaks. These are basically p-values. So this peak represents regions in which there is a strong association with the disease. So we found in this region a peak of association within the gene ataxin-1 that encodes for this protein that is with the same name, ataxin-1. So we were very interested in this region because ataxin-1 was previously known to be associated with a totally different disease. And this is why I started studying this gene. So one other thing that I would like to mention is that in this region, there is not only ataxin-1, but you can count on the bottom at least around 10 genes. So first, before moving with studying ataxin-1, we wanted to be really sure that the peak of association was really with the ataxin and not with these surrounding genes. So in our lab, we developed these statistical methods that can basically weight the association of the likelihood of each gene in the locus to be the real true variant. And as you can see in this heat map, we have each line is one gene in the locus, and then we have each row is a specific cell type. So we tested immune cells such as T cells, B cells, monocytes, but also brain cells. And this is all through publicly available data from NIH consortium. So all this epigenetic data set. We developed this statistical method to integrate this data set and develop these scores, which tells you which genes is most likely to be the real associated gene within the locus. And as you can see, ataxin-1 was the top one, was the most associated in all the cell types that we tested compared to the other genes. So this was a confirm for us that we could move on with the functional characterization of ataxin-1. And in general, for those of you who are curious, this understanding the real causative variant is a very common problem in GWAS studies, because we have a strong linkage disequilibrium in the human genome. So effects that are close to each other physically on the DNA molecule are very difficult to dissect. So this is just one of the most recent approaches using publicly available datasets, but there are many other mathematical and statistical approaches to try to solve this problem. So at this point, we were fairly confident that ataxin-1 was the gene to focus on. So as I mentioned, ataxin-1 was previously known, actually was first discovered in the 90s, because it's associated with disease that is totally independent from MS, that is the spinocerebellar ataxia type 1, also known as SCN1. This is a movement disorder that is caused by the selective death of cells in the cerebellum. There is this population of neurons that are called Purkinje neurons. They are here in red, that dies in SCA1 patients. And their death is caused by a mutation in ataxin-1. Basically, actually, ataxin-1 belongs to a bigger protein family that shares this domain that is a polyglutamine domain, this polyQ. So it's a stretch of glutamines that in healthy individuals, they span from 6 to 44 repeats. In SC1 patients, we see an expansion from 39 to 83. This mutation causes the aggregation of the protein, becoming toxic and killing these Purkinje neurons. This SCA1 is most basically a motor disease, but also has other effects on cognitive function. But nothing at this point, for almost 20 years, nothing was known about ataxin-1 outside the brain, the central nervous system. So we were basically trying to understand how this gene could be linked to an autoimmune disease. So we decided to go with an in vivo approach. And now with this slide, I will introduce what is perhaps the most common in vivo models to study MS. So MS is a human disease, so is not developed by animals naturally. But we found a way to introduce artificially a disease that can recapitulate many symptoms of MS. Basically, this model is called experimental autoimmune encephalomyelitis, or EAE, in which we immunize animals, mostly rodents, mice or rats, with short peptides from myelin proteins. After the immunization of these animals, the animals boost this autoimmune response against these peptides, and together with specific adjuvants to let the immune cells enter the CNS, they start a demyelinating disease. Typically, the onset is within the first 2 weeks. We observe like initial peaks around between 10 and 20 days. Then we see a partial recovery before entering this chronic phase. This is the most common model that uses as a peptide, the MOG peptide, that is a very common myelin protein. But playing with these peptides, such as from other proteins, such as MBP, PLP, or even other mouse strains, you can basically model other disease courses. For example, there are EAE models that have multiple peaks to model the relapsing, remitting. We have more progressive. So we use this array of models to try to understand the correlation between the immune response and the CNS pathology. So what we did was we obtained from a collaborator a knockout line, mouse knockout line for ataxin-1. So these mice are fine. They don't have any gross phenotypes. So we were able, so they could reach adulthood and they have no symptoms. So we induced EAE in these ataxin knockout mice. And what we observed was that the ataxin knockout mice developed a much worse disease, a much more severe disease compared to the wild-type controls. We also induce EAE in heterozygotes, and they also displayed a phenotype in between the knockout and the wild-type, showing some gene dosage effect. This more severe EAE course was also reflected in the higher mortality rates at the end point that reaches almost 75% for the knockout mice compared to the less than 5% in the wild type. So at this point, we could show an effect of ataxin-1 overall on the EAE phenotype. We were also interested to understand if the polyglutamine stretch that I mentioned before that is associated with the SCA1 was also playing a part in this phenotype. So we induce EAE in another ataxin mouse model that is used in the SCA1 field, in which scientists introduced this elongated stretch that caused basically many of the SCA1 symptoms. But in this case, we could not see a major effect on the EAE phenotype, except for a couple of days that shows a little bit higher disease score. So we could conclude that this novel function in modulating the EAE phenotype was not mediated by the polyglutamine score stretch. So after this, we move on. So I should mention that the ataxin-1 is expressed everywhere in the organism. So, and these knockout mice, they are basically full knockout. They don't have, they are constitutive knockouts. So at this point, we could still not narrow down the cell type and the tissue mediating this function. So to do that, we we employed another EAE protocol that is called adoptive transfer. Basically, you can transfer immune cells from an EAE injected mouse to a naive recipient. The naive recipient will develop still EAE, and in this way, you can introduce cells that are genetically different from the host. So we transferred ataxin knockout and ataxin wild type cells into wild type recipients. And we were able to reproduce the effect of the constitutive knockout, meaning that the deficiency of ataxin-1 in the immune system per se is sufficient to cause this exacerbated phenotype. So we could focus our investigation on the role of ataxin-1 in the immune system. So we didn't rule out the contribution of the CNS, but from this data, we could focus mostly on the immune system. That was a totally novel and unexplored function of ataxin-1. So how can we explain this basically protective effect of EAE or ataxin-1 on EAE? So I should mention that EAE is mostly driven by pro-inflammatory T cell subsets. In particular, there are two subsets. They are the Th1 and the Th17. They are the main driver of EAE. And there is also an opposite T cell population that controls the immune response. They are the Tregs. And I put in brackets, they are the molecular markers of this population. Basically, they are CD4 plus is a membrane marker, and they also produce interferon gamma, that is a cytokine, the Th1, IL-17 for the Th17, and the Tregs, they have characterized by this FOXP3 that is a transcription factor. So we could envision two different and still possible models. In the first one, there was a direct effect of ataxin-1 deficiency on CD4 plus on T cells. Or we could also envision an indirect mechanisms through the antigen presenting function. So to test in the lab these two possible models, what we did was to isolate splenocytes from wild-type and knockout mice, and then we stimulated these cells in vitro with the MOG peptide that we used to induce EAE. And then we basically measured the amount of each of these important immune cell population. And we could see that in the knockout cells in vitro, we saw a higher percentage of pro-inflammatory Th1, but no difference for Th17 and Tregs. So splenocytes are the cells that are isolated from spleens, and we know they have multiple population. We have T cells, B cells, monocytes, so we have a complex environment. But when we purified from splenocytes only the CD4 plus T cells, and we repeated the experiment, we couldn't see any difference between wild type and knockout. So we could conclude that there is the effect of ataxin-1 is not direct to T cells, but is most likely through an antigen presenting function. So our next experiment was to actually identify which cells can trigger T cells to differentiate towards a pro-inflammatory phenotype. So to do that, we employed flow cytometry, that is a powerful technique to look at multiple cell types and measure their subpopulation. And we look at two markers. They are CD80 and CD44. These are two very common activation markers. And then we tested multiple cell types for the expression of these markers. And we found that mostly B cells and monocytes knockout have higher level of these markers. In particular, we found that B cells also, they show higher level of these markers before and after immunization. So we decided to focus our investigation on B cells. We could functionally prove their enhanced antigen presenting function by performing co-cultures between B cells and CD4 plus T cells. And we found that the knockout B cells can trigger the proliferation of T cells at a much lower level of MOG peptide, showing that their antigen presentation function is enhanced in the absence of ataxin-1. So we decided to focus basically to characterize the B cell population in ataxin knockout mice. Here I'm showing this nice scheme from this Nature Reviews Immunology, describing all the main subset, B cell subset in the mouse spleen. Each one defined by this complex set of markers. So we develop a panel, a flow cytometry panel, to measure each of these B cell subset in the mouse spleen between wild type and knockout mice. And what we saw is that the specific B cell subsets, in particular B1A and B1B and marginal zone B2, they are differentially, their size is different between knockout and wild type animals. But we didn't find any effect for Bregs and follicular B2 cells, showing that the absence of ataxin-1 is causing some problems in the normal development and polarization of the B cell population. As a control, we also look at T cells, but we didn't find any difference in the main T cell subset, namely naive, memory, and effector T cells, corroborating the idea that the effect of the function of T cells of ataxin-1 is mostly on the B cell compartment. And this was also proved by the evidence that at the disease onset, we found a higher B cell infiltration in the knockout animals, in the CNS, but no difference was in the T cell infiltration between wild type and knockout animals. Still pointing at the idea that ataxin-1 deficiency mostly affects B cells in the context of EA. So after this first characterization, we look at other function at the effect of ataxin-1 on specific function of B cells. As probably everybody knows, B cells is the main cell types is the only cell types producing immunoglobulins, so antibodies. So we titrated the antibodies in the serum of these mice. So we look at two specific population of antibodies, the IgG and the IgM immunoglobulins. And we found that at the total levels of IgG and IgM were higher in the knockout, either naive or after immunization. We also look at the anti-MOG specific, so a specific autoimmune response against the MOG peptide. And again, we could find that the knockout animals had much higher level of anti-MOG antibodies compared to the wild type animals. But we didn't have any difference in the IgM. So showing that not only affect the B cell subset, but also B cell function. Then we also employ this new omics techniques, in particular RNA sequencing, to look at the gene expression profiles in knockout and wild-type B cells, to look at the genetic network that might be correlated with these differences. And we tested both baseline B cells at baseline or after MOG immunization. And we found that the knockout cells had genes connected to cell activation that matches what we found by flow cytometry. And after MOG immunization, we also found genes connected to cell cycle process. In particular, we found and we confirmed by qPCR two genes, the cyclin D1 and D2, that are key genes in the mitosis, are upregulated in knockout B cells, which correlates with the higher proliferation. In particular, when we tested for gene network, we found that most of the genes that are upregulated in the knockout B cells, they take part in a large network that is involved in cellular mitosis. And it's interesting because this is not a new information, because the role of ataxin-1 in cell proliferation has been shown previously in other contexts, in particular in cancer, in some form of cancer, and also in the proliferation of hippocampal neuronal precursor. So the idea that ataxin-1 controls cell proliferation is not novel. And from these previous studies, we also know which specific pathways are controlled by ataxin-1. In particular, we look at specific pathways such as the MAP kinases and the STAT pathways. Again, we employed flow cytometry based techniques that is called phosphoflow, that looks at the level of specific phosphorylated proteins in the cell cytosol. And we could observe that in B cells knockout, the upregulation of higher phosphorylation of specific MAPs MEP kinases like P38 and ERK. And we also found the STAT1 and STAT3 are hyperphosphorylated in the knockout cells, but also in CD4 and CD8. This is true both in naive mice and also after MOG immunization. So we could confirm that the same intracellular pathways mediating the proliferation of other cell types also involved in B cell proliferation. So at the level of at the molecular level, from all the CA1 studies in the last 2 decades, we know that ataxin-1 takes part in a large protein complex together with other proteins such as ataxin-1-like and capiqua. They form this large complex that sits on the promoter of specific genes and inhibits their transcription. So it's a negative gene expression regulator. So we were keen to understand if the same binding partners that we know that are involved in the central nervous system, they are also involved in mediating the function of ataxin-1 in immune system. So to test this in the lab, we decided to follow a conditional knockout approach. So we were able to obtain from a collaborator these conditional knockout lines for ataxin-1-like and capiqua. They are the 2 main interactors of ataxin-1. And we developed these 4 knockout cell lines in T cell specific and B cell specific. So we had these 4 knockout cell lines and we induced EA in all of them. So this is what we got. This is the result of all the EA experiment. And we found that only the deficiency of capiqua in B cells can recapitulate, at least in part, what we observe in the ataxin knockout mice. No effect was found for ataxin-1-like, meaning that capiqua must be downstream ataxin-1, while ataxin-1-like is not required. When we tested the T cell specific lines, we didn't observe any statistically significant effect, still showing that the function of ataxin-1 is mostly in B cells. So at this point, we could basically confirm that this novel immunomodulatory function, its main role of ataxin-1 in B cells, and we also found that the same binding partners that is involved in SCA1 also works in context of MS. So, but all this is basically was done in mouse. And basically, this is what this is basically the model that we came up with to explain the function that I just said. But still, again, this was all mouse data. We wanted to go back to human and trying to find a direct correlation between ataxin-1 and MS, the human disease. So we were lucky that another postdoc in my lab was doing a totally different study. She was looking at the DNA, these are very recent data. She was looking at DNA methylation patterns. That is one of the main epigenetic regulators in between MS and healthy controls in several immune cell types: CD4, CD8 T cells, monocytes, and B cells. And she found that in MS patients at the onset of disease, there is a general hypomethylation in B cells. So 1,000 genes are hypomethylated in B cells at disease onset. And what was interesting is that one of the most dysregulated genes was actually ataxin-1. In particular, we found there were 4 methylated regions that in MS patients were down hypomethylated, specifically in B cells. as it's interesting that these four methylated regions, they overlap with other functional loci that were defined by ChIP-seq, DNA-seq. So that means that most likely these regions are involved in the regulation of the expression of ataxin-1. And we confirm that the methylation, hypomethylation of these regions actually controls the expression of ataxin-1 in B cells. And we found that in B cells of MS patients at the onset of disease, ataxin-1 is actually upregulated. So if we go back to the model, we could think that ataxin-1 exerts a protective function to control the autoimmune response by regulating the APC function of B cells that control the polarization of this pro-inflammatory subset, such as the Th1 T cells. And the expression of B cells is regulated through specific circuits of epigenetic mechanisms, such as the DNA methylation. But could be that other mechanisms are involved. So I want to conclude with some future plans that we are actually testing in the lab. We are looking, because ataxin-1 shuttles between the cytosol and the nucleus, we are interested in understanding the role of ataxin-1 in the chromatin remodeling, and also to address its role in other APC cells beyond B cells, and also in the CNS. More in the future, we're also thinking to try to develop possible therapeutic strategies targeting ataxin-1 immunomodulatory function, and also to look at other autoimmune diseases in which ataxin-1 might be involved. So I would like to conclude acknowledging a lot of people that made this possible. First, my lab here at ECU that took over the project, and also all the collaborators that started this interesting story. My mentor at UCSF, Jorge Oxenberg, and all the division on MS at UCSF, and also collaborators are Baylor College, Huda Zogby. She was the first to describe ataxin-1 in SCA1, and also developed all these mouse lines that I presented today. And her postdoc, Q-Min-10, that now she's a PI at University of Alberta. And also from Harvard University, Nikos Patsopoulos, who helped with the GWAS study at the beginning. I would also like to thank the sponsor, the funding agencies, NIH, the National MS Society, and also the Italian MS Society. Thank you very much for your attention, and I will be happy to take your questions.
Thank you very much for your excellent presentation. You know, it's not our close research area, but the molecular approach is transversal to every area. So one interesting point is if you are planning to address this ataxin issue in the progression of disease, not only at onset, and what is the role it plays in this progression. You show is very different, the episode of remission and reset, but then in a second progressive stage, nothing to do. It's different, both stages concerning ataxin.
Thank you for your question. So we started because so the GWAS is about MS risk, is not MS progression. at the time when we started that experiment, the association was basically on MS risk. So there was that's why we focus on the initial stages of MS. Was recently published a few months ago, the first GWAS on MS progression. And what is interesting, that we found a totally different genetic signature compared to genetic risk. That means that the genes that control the risk of developing MS are totally different from the genes that controls the severity of the disease. And ataxin 1 is in the GWAS risk, but is not in GWAS on progression. This was totally new because was technically difficult to collect enough samples to do a GWAS on progression. So I could not exclude that there might be an effect on the brain and on progression, but I don't expect a major effect as on disease onset. But we are still planning to look at ataxin 1 in the brain in DCA mice, because we know that it's highly expressed in neurons, not only neurons, also in oligodendrocytes. So we are doing some experiment to address that, but based on this new data on GWAS progression, I don't expect a major effect, but it still could be possible because we see it's very complex disease. Could be the effect is there, but it's very small.
Okay, thank you.
Yeah, I can see you. I stopped. Let's see. Yeah, I stopped my presentation. I can see all of you. Do you still see my slides? Okay. Yes. Yes. Okay, no. So you take the spleen, the cells from the spleen. Yeah, so it's also possible to do a bone marrow transplant and then induce EAs. That's another model that you can use, but it's technically more difficult to isolate the bone marrow cells, and also you have to irradiate the recipients to kill the bone marrow cells in the recipients. So it's technically more demanding as a model. This model that I use is much easier. You take spleen cells and you just do an IP injection without any other intervention. And these mice will develop EA. Yes, but the cells that are injected comes from EA immunized mice. So only the cells that you inject are to pro-inflammatory phenotype. So the wild type cells in the host, in the recipient, they will not contribute. So I know it's not as perfect model. It's a little bit dirtier than a bone marrow transplant model. We are planning to do that. But then COVID hit, we had to stop all research at UCSF. We had to stop all the breeding, all the mouse lines. So yeah, we had a big change in plans in 2020. So I hope here to develop more refined models to look at the role of ataxin-1 in specific. So we are developing a conditional knockout line for ataxin-1 that we can use to ablate ataxin-1 in specific cell types without doing this adoptive transfer.
I have a question.
Yes.
Nice presentation. Congratulations.
Thank you.
You mentioned that one of your students discovers the hypomethylation of B cells. In those patients, you also demonstrated the hyperproliferation of B cells, or you only checked the hypomethylation?
That project was, so the amount of cells was very limited to do functional studies. Those samples were collected within 12 hours from the first diagnosis of MS. So we had only a few million cells to do the DNA methylation profiling. So we didn't collect any functional, we didn't have enough cells to do cell proliferation. It was very, very difficult to do that. But we could find, we did RNA sequencing on those cells. We could find genes associated to proliferation. But yes, we didn't it was very difficult to do a follow-up on that.
Okay, thank you.
Thank you.
Sorry, Vicky, if there is no more questions, I want to particularly thanks Alessandro to the excellent seminar lecture you give to our group. And it's a very nice to hear foreigner researchers. So for us, it's a pleasure to share this morning for us with you.
Thank you so much. It was my pleasure to present my research. We can think about to do the opposite, that you present your research to us. I would be happy to have the same here.
Okay.
Okay. Thanks for the invitation.
Thank you.
Thank you so much. Have a good day.
Thank you, Alessandro.
Bye.
Bye.
Bye.